Okay, let's try this all again. So now that I have my little highlighter. Okay, so the idea is you should have a sterile loop left over. If not, you can always kind of replace it with a Q-tip from home as long as it's clean. Like I said before, we're going to do that fishtail pattern that everybody knows, right? You're going to do as large as a fishtail pattern as you can on all three plates that you have. The darker color EMB, the lighter color MSA, and either the TSA plate that you had previously that you created in the lab or one of the ones we gave you. And then keep those guys in, again, a warm place out of reach as usual, and save those out for at least 3 to 4 days tops. And then if they're still overgrowing, you can always put them in the fridge if needed. As a reminder, again, make sure you take care of the live sample of the E. coli that you're going to be taking. And again, you may discard the EMB and MSA plates once they've grown and once you've actually taken your samples, but do not discard the TSA plate. So I'll show you a little bit of an example of that in a moment. So here's what we have. You should have 3 plates ready: a light red version, which is the MSA plate, a darker red version of it, which is your EMB plate, and then again, a TSA plate that you have made, or again, the spare one that we just provided you with in the restock, just in case, right? Now in the restock, for those of you who remember, you should have had 2 plates, one and the vial that includes the E. coli. Now, one of those plates is actually marked with a little black dot. This is a Mueller-Hinton plate or a Hinton-Mueller plate, which is specialized for antibiotic resistance. So that's actually a relatively expensive plate, so please don't mess with that one. And so we're going to be using that later on in a couple of weeks when we set up the last set of experiments. The other plate that you should have that looks almost identical to it is just a regular good old TSA plate. That's the spare one that I'm talking about. So in the event that you didn't have your original TSA plate remaining at home, you can use the spare one as well. And then in there, you should have your E. coli vial, again, emphasizing that you should treat this guy with respect. It is a live sample. And then you should have in a tiny, tiny little envelope a bunch of sterile filter discs that we're going to use for the antibiotic resistance, and the paper matrix that you're going to be using for the second unknown later on. So what's the plan with this? All right, well, then the idea is, let me see if I can change the colors over here, is that we're going to take our little vial of E. coli, reminding you again to be very, very careful with this guy. It could be relatively full. You may need to kind of resuspend the sample inside of it. And you're going to want to kind of pop the cap open, right? And this cap is fairly well sealed. It's designed so that nothing spills. And so when you pop it, just be careful, kind of, so be very gentle when you're opening, as opposed to you want it to spill it all over the place. And so you're going to take either your loop that you should have, one spare one, or if you don't, find a little cotton swab that you may have at home, right? And you can use that to create your fishtail. And so take each time one little bit of sample and transfer it to each plate. So each plate, you're going to create a fishtail each time, right? And so you'll do it for the MSA plate and you'll do it for the EMB plate altogether. And once you're done with these guys, then again, you're going to want to incubate them for about 48 hours to 72 hours, right? So about 2 to 3 days, and then observe the growth. And the idea behind this is that you're going to want to compare. The concept is, how are these 3 plates growing this particular sample of E. coli? Do they grow them the same? Do they grow them differently? That's what you're going to want to include in your lab manual and your lab notebook. That's the overall gist, right? So the setup for today is you want to inoculate all 3 plates, set them up in a warm area. Again, you need your results for your next session, depending on which session you're in. And then by that time, make sure you take your pictures so you can compare them, and you're more than welcome to discard your EMB and MSA plates. However, do not discard the TSA plate even after growth, and do not discard your E. coli vial in this case, because we're going to want to keep them for a couple of weeks so we can use them for labs 42 and 43 later on. That's what we want to do. So for this, we're going to want to place these inside a little baggie, inside a little Ziploc. I'll show you in a moment how we want to do that, right? And put them in the fridge so they can last a little bit longer for the next set of experiments. That's what we want to do. So why we're doing this kind of comparison issue is kind of what we started with. The purpose of this lab is that overall each lab in the metabolic labs 19 through 38 is to try and understand why we actually do the experiment in the first place. What does it accomplish for us and what do we learn? And more or less what this is doing for us is classification. So when we did our pre-labs for this, we asked you to give us 3 things. Same as always, the objective, the purpose, and the methods kind of thing. They've been tweaked out a little bit like we showed you in the previous videos. And so the idea is, well, tell me when I ask you, hey, what type of analysis is being performed in this particular lab? Your answer, your conclusion, your analysis behind this is to be able to tell me whether these tests are being selective, whether these tests are being differential, or they're being possibly both, that we do have some tests that combine both abilities. But then at the same time, I want you to tell me about the metabolism. Like I said before, that whole idea of tell me what you eat or you are what you eat kind of thing, right? This is what we're trying to find out. Their metabolism, what they're processing inside that medium, can tell us a little bit about their taxonomy. So usually it's either some sort of pathway or at least some sort of enzyme being used in that particular analysis. And so your job is to be able to tell me either the key enzymes being used or the key materials being used and how those reactants and products are taking place, right? But not only that, you're going to actually have to include specifically how is the test being performed. Now, most of the time, there's basic things associated with them, such as, we're going to incubate it for, 48 hours. We're going to put them in 37 degrees. Those are all pretty standard, unless there's something unique about that protocol. But the parts that are different are the ones I want you to be able to tell me. So were there any specific ingredients that we needed to add for this test to work, right, that are different from all the other tests? Or perhaps they required a longer period of time or a shorter period of time. Maybe it required only 6 hours, or maybe it required 4 days. So you need to be able to tell me that. Or if there were any changes in the incubation temperature. So instead of being at 37, which always mimics body temperature, did it take, only 20 degrees or something like that? So you should be able to tell me these things as well. And then once you complete your incubation period, did you have to do anything afterward? Are there any post-assay steps? In other words, once you waited for your 2 or 3 days, did you need to add anything? Did you need to change anything? Did you need to heat something? Did you need to mix something? You should be able to tell me that information as well, right? And so then we have the last little piece here is what type of results we're going to obtain from those assays. And so in this case, yes, normally there are positive and negative results, which is what we expect, but sometimes there are alternate results as well. So you should be able to tell me what all of these results are possible. The good ones, the bad ones, the middle ones. It's never just kind of a yes or no. Most of the time it ends up being a little bit of a spectrum. Certain things can happen in the middle in there. But not only that, sure, you can tell me yes or no, but you're going to have to also tell me what does it mean. When you get a yes result, when you get a positive result, what does it mean in terms of the test? So what did you learn from that particular example? But the same thing applies for the negative results or any other optional results that you can get. So let's back up a little just to kind of summarize what we've done and what we're doing specifically with the lab that we just set up. And I'm going to show it to you on the screen. So for example, today we're setting up lab 19, at least the remote version of it. And so you need to be able to tell me what lab 19 is doing as far as the EMB plate and the MSA plates, for example. Are they selective tests? Are they differential tests? Or are they both? In other words, do they kill one thing and leave the other one alive? Or do they kind of just change in colors or in certain types of morphologies? Or do they do both? That's what you need to be able to tell me about lab 19. Then you're going to also tell me what's kind of going on in terms of their metabolism. What is actually happening in terms of the organism and what is it doing while it's inside that medium? And then, as always, you should be able to tell me the minimum key methods that are involved in this. Now, for lab 19, it's kind of easy, right? We're only just using 2 plates. There was no additional crazy instrumentation or specific media or tubes or instruments that we were using in there. But you do know that, for example, the MSA plate, the lighter version of the 2, its name kind of gives it away a little bit. It has key ingredients in there. It's called mannitol salt agar, right? So in this case, that medium has sugar and it has an extra high amount of salt in it. And so that's kind of what you should be including. That's what you should be learning for that particular test. Now, at the end of this one, we're not really adding anything, so that didn't need to exist for this particular test. But later on, you're going to have to add some reagents. You're going to have to add some drops of certain things. For example, in the hydrolysis test, when we do like the starch test, you're going to have to add iodine at the end. Or whenever you're doing certain incubation periods, you add some drops of certain reagents to kind of see what happens. So that's kind of what I would want you to be able to tell me here. But then you also want to be able to tell me about those results. So in this case, in the EMB and MSA tests that you're performing, what does a positive test look like? What does it mean when somebody actually grows or it doesn't grow, a negative result? Or what happens when something grows and does something slightly different? So you're going to be able to tell me the meaning associated with those, right? What does it mean when you get a positive result? What does it mean when you get a negative result associated with them? Okay, so we'll go back to this kind of setup over here and kind of talk about what we're going to do. So I'm going to actually show it to you with the actual plates on the screen. And so I can show you kind of the overall plan using the E. coli, the live sample, your sterile loop, that kind of thing. And then we'll let you guys perform the experiments themselves. So let's show you more or less how we want to see this kind of live. So we're going to take off the presentation so you can actually see the actual setup for this. And so in the new kit that you guys all picked up, you picked up a set of 2 plates, right? It had a little rubber band around them. And I want you to notice that there's one key difference inside of them, in which one of them had this tiny little dot placed on them. That's the Mueller-Hinton plate. So I will encourage you guys to at least kind of mark it up a little bit. That way you know that that's the plate that you're not supposed to touch for another couple of weeks. So this one's a specialized plate. And even though it looks almost identical to this guy, leave this guy alone. So save this guy somewhere else, put it back into the fridge. This is going to be your spare TSA plate. So again, be aware, little dot, that's what makes it the difference. So that's a specialized plate. Also in there in your new kit, you should have received a little vial that again should have this little Eppendorf, has this little kind of sealed cap next to it. And it's kind of coming out a little bit blurry. And so you're going to need to kind of resuspend it a little bit just so you can get the organism to kind of spread out in the fluid. I'm going to save it over here. And then everybody should already have their series of plates that are the EMB and MSA plates. And so in the previous kit, you probably had these guys. And some of you may have slightly larger plates. It's okay. That makes no difference whatsoever. And so you have 2 key plates. You can always tell the difference. The lighter version of them is going to be your MSA plate. The darker version of them is going to be your EMB plate. That's how you can always tell them just by simple kind of tones in the actual sample. So these are the 3 plates you need. And so you should also have remaining 1 of your loops from your sterile loop kit, right? If you don't, again, go find a clean Q-tip and that'll still do the trick. So we're going to take our loop. You already know these are sterile loops for us. And then same pretty simple idea. So this is the part that I wanted to demonstrate carefully. These little Eppendorfs kind of are a little finicky. And so this cap is well sealed in there. And so the idea is I don't want you to pop it accidentally and, hit your face kind of thing. So again, goggles, mask, gloves, all the good stuff. So be very, very gentle when you're trying to pop this guy open. All right. And then minimize the amount of time that you have it open. So kind of dip this guy in there. I'm trying not to crash with the camera over here. And you already know what to do. All right. Take your plate, do a little fishtail, and that's it. That's done. And you're going to do it for all the remainder of the plates. So you can actually kind of redip really quick. All right. Close this bad boy up. Open up your plate, little fishtail. Pop up, close, little fishtail. And then you can discard your loop. You can hit it with H if you want to, but that's it. All 3 plates are set up. And then we're going to give these guys at least 48 hours to 72 hours to grow. And then if they, for whatever reason, start overgrowing, put them in the fridge so they can slow down. So you want to take a picture now so you can know what the plates look like before growth started to occur. And then you're going to take a picture after 2 or 3 days, so you can see what's going on. Now, once you're done, you can discard these two. You can get rid of them as usual. But this guy, you're going to want to save. And so you're also going to want to save this bad boy over here. So what you're going to do is find a little Ziploc. And even use some of the ones that we've had before. Put your plate in there. Again, after incubation, not before right now. I'm just kind of demonstrating here for the sake of just showing you what I want you guys to do. So after it's done growing, so after you see stuff on here, put it inside a Ziploc. You can put this bad boy in there too. And I normally have kind of paper towels kind of spare around, running around. So I'm going to take, good old paper towel. There's nothing amazing about this guy. I'm going to kind of cut it into small little pieces. And normally, I can fold it up a couple of times. And just this little chunk of paper towel, add some water to it, moisten it. It doesn't need to be sterile, just a little tap water. And then you're going to put it inside the same kit so it can add moisture to this area. And then seal this bad boy without fully closing it. So you'll notice that I'm going to leave just a little bit of a opening over here. So kind of little opening over here, right? So it can kind of breathe per se. So it can use a little bit of oxygen in there. And so this guy you're going to want to put inside your fridge for a couple of weeks. So it'll stay kind of nice and even without growing until we use it for the next experiments. That's the overall plan. Okay, so let me go back to the previous presentation to kind of show you what we're supposed to be doing with this. So you should be able to see the setup again, right? And so we took 3 plates, the spare TSA, either that you made before or the one we sent you with, again, not the one with the dot, that's a Mueller-Hinton plate. And then the MSA, light red, the EMB, dark red. All 3 plates get a fishtail. Wait for a couple of days, see what happens. After you have evidence, after you take pictures, you can discard these 2, but save this one and the E. coli inside a little baggie and put those in the fridge. Again, you're going to put these in the fridge after they're done growing, not before. If you put them in the fridge before, nothing's going to grow at all. So give all 3 of these plates a chance to grow. And so you'll need growth by the next session that we meet. So you only need 2 to 3 days, maybe 4 if it gets a little bit too long for them to grow. So you can plan your time accordingly. So that's pretty much the setup that we have for these labs. Again, as we come back next week, we'll be able to also discuss kind of the meeting of what those results show you, whether they're positive, negative, or somewhere in the middle behind that.